if possible, I don't know, I haven't attended these in the past, but feel free to interrupt with questions, or I think the more interactive this is, the more exciting it could be, or we could do questions at the end, whatever you guys have done. But I'm totally open to questions throughout. So I'll introduce this talk really by talking about some of the classical genetic mouse models of small cell lung cancer that have some of the limitations in making these models more complex. And this really leads into more higher throughput, easier to use CRISPR-based mouse models, which have advantages and disadvantages. But one of the major advantages is the ability to target additional genes. And we know a lot more about the complexity genetically and epigenetically, transcriptionally in small cell lung cancer that we've been appreciated over the years. And so this is I think, an easier way, at least, to start to capture and understand that. And then we'll talk about really increasing the throughput of these CRISPR-based models through screening, so work actually Julian Sage's lab has done. And then some alternative approaches we'll go through, such as syngeneic allografts or pre-SCLC models as an alternative to doing these very rigorous autologous mouse models. And then just want to share some ideas about some limitations, but also where I see the future going here. So as you all know, being in the small cell lung cancer field, we have appreciated really over many, many years, this was work from Julie George in 2015, that small cell is really universally characterized by loss of function mutations in RB and P53. There are a few exceptions that appear to have RB wild type tumors, but broadly, this is both RB P53 loss. And then the whole genome sequencing or exome sequencing has identified several other mostly tumor suppressor genes with loss of function mutations, but these are not nearly at the level or frequency as RB of P53. And then conspicuously, we really haven't identified many recurrently mutated oncogenic drivers. This is in contrast to non-small cell. So with the knowledge, and this is about 21 years ago now, with the knowledge of this universal RB P53 inactivation, Antoine Burns's lab developed the RP model. So this was published in 2003, and this is basically the first genetically engineered mouse model of small cell lung cancer that was developed using conditional flox alleles against RB and P53. And so adenoviruses, adenocre, was intratracheally introduced into these mice that harbored these flox alleles for RB and P53. RB and P53 got deleted, and they started to see tumors form. The first tumors were observed around 210 days after the intratracheal injection, but on average, it took about 12 months for tumors to form and mice to succumb to the disease. And so this was a really long latency period, and people, I think there's been an interest over the years in different studies. Why is this latency so long? We know, different mutations can accumulate, particularly copy number alterations. I think it's still a big unanswered question of why, when you introduce these truncal events, there's a year delay in tumor formation. But that won't be the topic of this discussion, but just to point that out. And then other investigators over the years have introduced additional either flox alleles or transgenes to modify this model. And I think two of the, I'd say, standard models in the field now are using what's called the RPR2 model, which was initially developed by Julian Sage, where RBL2, which is mutated in small cell lung cancer less than 10%, but it's another pocket protein in the RB family that was introduced along with RB and P53. And that decreased the latency from about 9 to 12 months to 6 months. And many more lung tumors formed in the RPR2 mice compared to RB P53 mice. And then Trudy Oliver's lab put in a transgenic MYC into the RP mice. And this not only accelerated tumor development more to like 2 or 3 months, but also changed the phenotype associated with those mice, and we'll go into that a little bit here actually. So the classic RP model and the RPR2 model both form histology, I guess, that we would define as classic small cell lung cancer. Now we know that this is more associated with ASCL1 and the ASCL1 subtype. And when Trudy's lab introduced MYC into this model, they observed a more variant histology. And the variant histology in cell lines, at least, had been described in the 1980s, that they don't form as spherical neuroendocrine spheres, but more clusters of cells, and some of the cells attached to the plate. And these is what John Minna and others called the variant histology. But the variant histology in mice is associated with more a neuro-D1 subtype. And Trudy's lab in subsequent papers has shown that these undergo lots of plasticity, not only to form neuro-D1 positive tumors, but then to evolve into more non-neuroendocrine states. And so I think classically, really, these are the three classic genetically engineered mouse models that the field has been utilizing. And this has led to actually many powerful insights on small cell tumorigenesis, growth behavior, metastasis behavior. But I think one of the major limitations in these initial models is just how complicated it would be then to introduce an additional target gene of interest. So you can imagine you can breed these mice, and that's what they did. Julian did, and Trudy did with the RPR2 and RPM models. But you're always dealing with these initially 4 flox alleles, RB and P53. And so you can imagine, if you want to introduce an additional target gene of interest, if the conditional flox mouse exists, you probably have at least a year lead in to breed those mice. So this is just true of any classical genetics. And then the other thing is, we were appreciating at the time, this is now about 10 years ago, that there's many other mutations in small cell lung cancer, but they're at very low frequencies on average. And so really, if you want to kind of cover, potential tumor suppressors in small cell that are otherwise mutated with RB and P53, it's a large project for each one. So I think there's evidence of this from the initial Cancer Cell paper in 2003 to now, or to last beginning of this year when we published this. There was really only about 20 or 30 other mouse models where investigators had incorporated additional alleles. And this, I think, speaks to how complicated it is and burdensome it really is, a whole postdoctoral project, and a very involved postdoctoral project to get to the point of even characterizing one model with one additional gene. So I encourage you to, with Julian, Quan, David, and Trudy, we wrote a review on all of this. So I'm not going to go through each model and finding here, but you can find all these tables in the paper that, at least to our knowledge, was comprehensive by the sort of mid-2023. And so I kind of alluded to this, or directly stated it, that really the challenge here in these models were, it takes roughly around at least 3 years and a very, very involved project, really just to test one other candidate gene, breeding these into these flox alleles. So really my question, and this started actually when I was starting my postdoc about 10 years ago, and I'll show you how this is inspired, but can we do this better, faster, and easier? And then can we start to model some of the complexity of either genetic co-occurring mutations or inter and intratumoral heterogeneity? And so now we appreciate that not only is there genetic diversity of small cell lung cancer, there's massive transcriptional diversity of small cell lung cancer. And so I mentioned that the RP or RPR2 models are really modeling a pure ASCL1 subtype. And what I'll get to is that not only are there these distinct subtypes of small cell lung cancer, actually there's a lot of intratumoral heterogeneity that we observe in human small cell lung cancer. For example, ASCL1 and NeuroD1, although presented here in the initial review as sort of distinct subtypes, and they are, usually one is dominant or the other is dominant. There really are a large number of these. You can even appreciate on the RNA-seq data that express a little bit of the other transcription factor. And Charlie Rudin and Marina Bain have shown this by IHC as well in human tumors. And then, as you'll appreciate, I mentioned the MYC transgenic models the NeuroD1 subtype with some non-neuroendocrine evolution, but we really don't have a good model for these other two subtypes at all, COT2F3 and the inflamed subtype. So these are also just open questions. Okay, so now we're going to switch gears to, can we do this easier with an all-in-one CRISPR-Cas9 approach? And so when I was thinking about this, this was actually 2014. I was kind of beginning my postdoc in Bill Kalin's lab. And from Feng Zhang's lab, Randy Platt came out with the really first transgenic Cas9 mouse paper. And in this paper, there were several sort of proof of concept experiments, but one of these was that he had developed a KRAS LKB1 P53 model. And this was, I think there were challenges with this model, particularly related to incorporation of the KRAS mutant allele. But I think it was a proof of concept of endogenous gene editing where you can include multiple guide RNAs in one AAV. They're using an adeno-associated virus at the time, and form lung tumors. And so, I thought of this as I was thinking through projects. And so I adapted this really, you can see it looks a lot like the construct that Randy had generated, basically taking the RPR2 model, finding guides that could edit RBP53 and RBL2 or P130, including CRE, a luciferase. And then basically it's an all-in-one AAV construct that then we could show we could delete all three genes in cell lines. This is in nephs that express Cas9. And then we introduce this into these lock, stock, locks, Cas9, P2A GFP mice. Initially, actually, they were mixed background, and now we use pure black 6 for this. And then what we saw, we basically tracked tumors over time. And basically around 8 to 9 months after intratracheal injection of this all-in-one AAV, we started to see most mice form tumors. And the latencies is somewhere between 8 and 10 months. And by 12 months, almost all mice succumb to their disease. So these are just some examples of some of the original mice that we were imaging over time. I would say, we have tried to adapt this to luciferase. This wouldn't work for the previous construct because this doesn't integrate, but we've tried to look at endogenous luciferase reporters. Been very challenging to use anything other than MRIs to really track these mice over time. There's a lot of false negatives with the luciferase in our experience. And so from this first cohort, we characterized the tumors, and these were neuroendocrine tumors that were ASCL1 positive, synaptophysin positive. Most, as you'll see on the table at the bottom, most of these tumors metastasized spontaneously to the liver, and then we saw some kidney adrenal mets. So this recapitulated, what had been reported for the RPR2 model, very similar histology, similar subtype. In our second cohort, we appreciated actually with this AAV that we developed, some mice developed histiocytic sarcomas, which we think was actually an off-target effect of the broad tropism related to the AAV. And so we actually switched to adenovirus. This was what was being used in the classical genetic models. And now really the incidence prevalence was all small cell lung cancer, close to 90%. And so we could characterize these initial tumors. This is just CRISPR amplicon sequencing. So we're basically taking the amplicon where we've targeted RBP53 and RBL2, and we saw really in every single tumor there was selection for complete loss of RB and P53, which you'd expect based on the initial mouse model, but that was encouraging to see. And then RBL2, there was always deletion or an indel, but not always complete loss of RBL2. So this is one difference from the RPR2 model, where Cre-Lox-P recombination is always going to delete both RBL2 alleles. Here, sometimes we basically get what's selected for, and I think part of this is it's a little more inefficient system, and I think that actually has potential advantages in terms of gaining insights into the biology. So sorry about that. Okay. Yeah, and so similar to the RPR2 model, these tumors really homogeneously expressed ASCL1 and not any other markers of different subtypes. Here's just showing NeuroD1 as well. I think one interesting aspect of this model that we've taken advantage of now is single cell sequencing technologies come into play. So this was a little later on, is that the endogenous Cas9 transgene is linked through a P2A to GFP. So you can actually sort out these tumor cells, and you can do this very early on, or actually in established tumors, and really analyze just the tumor cells themselves. Or you can look in single cell RNA-seq as markers of the tumor cells to sort of confirm what you'd expect with ASCL1 expression, but the GFP and Cas9 can be quite helpful there. Okay, so as I mentioned, really the first generation genetic models, both the classical and really just this CRISPR-based model as well, don't really fully recapitulate the transcriptional heterogeneity or even the genetic complexity of small cell lung cancer. And so the next question here, and this is really why I went to all the trouble of really at this point remaking the RPR2 model in a CRISPR-based fashion, because this has the opportunity. Then the thought was, can we introduce additional target genes of interest by simply just cloning a guide RNA, just like you would in the lab? If we do CRISPR cloning, it's very simple, ligation cloning, traditional cloning. We basically made these vectors so we could do this with this complex adenovirus. And then we made this, it's through a gateway system, so it's through a destination vector system, that then we can make these custom adenoviruses. And the idea is then we could study either different tumor suppressors that were rarely mutated in small cell lung cancer or mutated at low frequencies. We could also study different drivers that we think could be good therapeutic targets in small cell, and ask what happens when we delete these at small cell tumor initiation in this autologous model. And then the hope, although this is more of the hypothesis, can we make models that could capture different subtypes of small cell lung cancer and some of the transcriptional diversity and heterogeneity that we now appreciate in the human disease. And so let's see. So we initially did this at the time. I was still finishing my postdoctoral fellowship. We were interested in this histone demethylase KDM5A that interacts with RB. And we had in vitro data to show that there's an epistatic relationship between RB and KDM5A, that when RB is lost, cells become dependent on this KDM5A histone demethylase. And so we wanted to see what is the role of KDM5A in small cell tumorigenesis, but also characterize this as a proof of concept. Can we really understand how inactivating an additional target gene of interest may play out in this model? So as I mentioned, when we switch to adenovirus, really almost all the mice get small cell lung cancer. And we could, characterize the mutations in these mice. Here's just looking at RB, P53, and P130, and then also the additional target gene of interest. So in this case, it was KDM5A, and we could use CRISPR sequencing to look at KDM5A, and only the KDM5A knockout mice deleted KDM5A, and the others retained it. So in the KDM5A is important for small cell tumorigenesis. We found that when we knock out KDM5A, tumorigenesis is delayed, but tumors were actually able to form in the absence of KDM5A. So all the tumors had these loss of function index. And so I think there was plasticity associated with this model. We didn't really characterize it thoroughly at the time. But one thing, and I think that's not the point here, but the point here is some oncogenes, there'll be bypass mechanisms where they can lose the oncogene of interest and develop a different phenotype to form tumors. I'll show evidence of this today. We have an exciting story, hopefully, that will be coming out in the next 6 months or so, I think, that really displays this. But alternatively, there's oncogenes that are essential for small cell tumor formation. And so we had tested ASCL1 in this model. Jane Johnson's lab had elegantly shown that when you delete ASCL1 in the RPR2 model, the traditional model, really no tumors form at all. And so we wanted to test that as more of a proof of concept in this model. And what we found was quite interesting. And this gets us some differences between classic genetic models where the gene itself is always going to be deleted. And then you can analyze the phenotypes associated with that. Whereas in these CRISPR models, you can look at selection, in this case, ASCL1, to get at really how critical is that gene and what do the cells do to try to bypass this gene. So what I'm mentioning here is this panel in D, where these mice got this ASCL1 guide, and we can see basically that no tumors formed with this ASCL1 guide, forming a frameshift deletion. But what we found in all the tumors that formed was either not small cell histology or small cell histology, where we get these very rare in-frame deletions that basically select for retaining ASCL1 function. So CRISPR, most likely, and this is why you get knockouts with CRISPR, is the highest frequency event is a single nucleotide deletion or insertion, and in-frame deletions are incredibly rare. So the evidence that we see a 48 nucleotide deletion, a 12 nucleotide deletion, and a 57 nucleotide deletion really speaks to the and ASCL1 was targeted right at the end of ASCL1. This really speaks to that the tumors that formed needed to restore ASCL1 function, and this looked very different in cell lines. I think one difference of these models is actually you can interpret sort of selective pressures for the gene of interest really by looking at the mutations that form within that gene. And so we wanted to use this to study this gene KDM6A. And KDM6A is mutated in about 3 to 4% of small cell lung cancers. And we thought it was interesting as well because it binds to KMT2D, which we know is really the third most frequently mutated gene in small cell lung cancer. So it forms a complex with KMT2D and 2C. And then it's implicated in cellular differentiation in other lineages. So the hypothesis really was this could have an effect on either lineage plasticity or neuroendocrine differentiation. And so what Leslie and Yixing did in my lab is they deleted KDM6A or kept KDM6A as wild type using non-targeting or intron targeting guide RNAs. And we can see that we can make these adenoviruses where all 4 genes were deleted, or just 3 of the genes were deleted, which we'll call the KDM6A wild type tumors. And so initially, actually, we're quite, this was during the pandemic, slightly after 2020 when we started in the lab again. Well, we initially were quite disappointed. The mice started getting sick, and it didn't seem like there was strong selective pressure for KDM6A loss versus KDM6A wild type. But then when we started looking at the tumors that formed after KDM6A loss, we appreciated this striking phenotype where tumors that were KDM6A wild type were all strictly ASCL1 positive and didn't express NeuroD1, and the KDM6A mutant tumors heterogeneously expressed ASCL1 and NeuroD1. And you can see there's a lot of heterogeneity even between the phenotypes in the tumors. So we can talk a little bit about that. And so characterizing these tumors more, we looked first at RNA-seq, comparing all the KDM6A mutant tumors to wild type that I'd shown on the previous slide. And really some of the top upregulated genes and gene signatures were NeuroD1 gene signatures. And when we looked at the single cell level, so this is just multiplexed IF and IHC, what we found was that this was a mutually exclusive expression of ASCL1 and NeuroD1 in different cells. So doing further epigenomic characterization, this is with ATAC-seq, we could look at the changes in open chromatin between KDM6A mutant and KDM6A wild type tumors. And what we saw was consistent with our expression results, that the promoters at NeuroD1 were all open in the KDM6A mutant tumors. And I think what was striking actually was when we did motif analysis and looked at the top motifs that were then open in KDM6A wild type tumors, NeuroD1 was the very top hit. So it wasn't just that there's lots and lots of different changes, although there are when you mutate an epigenetic modifier, but there is very strong signatures associated with NeuroD1 itself. And NeuroD1 was one of the top upregulated genes. So then to get at what are the mechanisms really by which KDM6A is driving this, we started to think about, okay, well, what does KDM6A normally do in cells? As I mentioned, it forms a complex with the compass complex, where it binds KMT2C or KMT2D and enhancers. And this complex together promotes H3K4 monomethylation enhancers, allowing for open chromatin. It also has this dual function where KDM6A demethylates H3K27. So together, what this does to chromatin, it makes it in a more permissive state for gene activation. So KDM6A function is normally associated with gene activation. So the hypothesis is this would normally maintain the ASCL1 subtype in an ASCL1 positive tumor. And so we started to look really genome-wide to analyze chip data of KDM6A chip and enhancers. And what we found that consistent with our hypothesis, KDM6A was most highly enriched at ASCL1 targets across the genome compared to really all other genes. And then when we compared H3K4 monomethylation enhancers in KDM6A wild type versus KDM6A mutant mice and H3K27 trimethylation enhancers, we saw the changes that we'd expect associated with KDM6A loss. So the H3K4 monomethylation enhancers went down, and this was most strongly significant at ASCL1 target genes. And the trimethylation H3K27 went up. And so this at least correlated very strongly with what we'd expect as a direct effect of KDM6A. So this part of the story, I think we can conclude that KDM6A has these two functions in small cell lung cancer. And in an ASCL1 subtype, this is keeping cells in an ASCL1 positive state. So the question then became, what promotes this NeuroD1 expression? And so, NeuroD1 goes up, it doesn't go down. So we hypothesized this was an indirect effect. And so we started to look at what genes may be upregulated in KDM6A mutant tumors in the tumor cells themselves. And then we asked, out of these significantly upregulated genes, surprisingly, were not that many, which of these could actually bind using chip data in NeuroD1? This was using publicly available chip data on the system. And then which of these could actually regulate the accessibility peaks that we saw in our ATAC-seq? And actually, things don't usually work out this way, but there's actually only one factor that was upregulated at that point. And that was KMT2A, which is part of a MLL complex, but more promoters than enhancers. So different than KMT2C or 2D. And what we found treating cells really unbiasedly, we did both of these panels in parallel with different epigenetic inhibitors that normally promote gene activation. So all these different inhibitors normally promote gene activation. We want to know blocking which of these targets, which I'll tell you, VTP 50469 blocks menin KMT2A, could downregulate NeuroD1. So I think coupling this correlative data with the functional data, this suggested KMT2A was somehow binding NeuroD1 and driving its expression in the absence of K6A. So we collaborated with Naomi Olson and Scott Armstrong's lab, who really are experts in menin, and they did menin chip. And what we could see was that when we chipped menin at NeuroD1, it was decorating the NeuroD1 promoter, and this signal was lost when we treated with the inhibitor. So in the KDM6A mutant cells, menin now has sort of a gain of function activity that's binding NeuroD1 and activating it. And so this was the conclusion from this study, that really KDM6A is driving cells in an ASCL1 positive state, but when you lose it, there's this gain of function activity of KMT2A and menin, and this drives this ASCL1 NeuroD1 heterogeneity. And so, I think this, together with the MIC model from Trudy Oliver's lab, models different aspects of NeuroD1 small cell. Our model is still, I'd say, more of an ASCL1 dominant model, where we see gain of NeuroD1, but it retains ASCL1, where MIC is such a strong oncogene, it's really driving NeuroD1 in a non-neuroendocrine phenotype. Okay, so I think this was just an example of, I think, what we can do with the all-in-one CRISPR-Cas9 approach. So now, do I have till 1, Ben, or? Yeah. So now I'll talk about some other higher throughput approaches for gene targeting. And this right now, has been pioneered from Monty Winslow's lab and used in Julian Sage's lab to screen for in higher throughput for phenotypes associated with CRISPR knockout somatic editing. And so this approach is called TubaSeq, which really stands for, like, basically we're sequencing barcodes that are introduced into guide RNAs. And so, different from the techniques that I've mentioned, we can use a pooled approach, just like CRISPR screening in vitro, although a little more complicated, but a pooled approach to really deconvolve after tumors form what genes may drive a phenotype. And Julian's lab has looked so far more at tumor growth. But I think one big difference between these models, which probably creates some challenges, but the Sage lab has been able to overcome this, is any approach like this with CRISPR screening, whether it be in vitro or in vivo, requires integration of the virus. Unless you're doing whole genome sequencing and really sequencing all the indels, which I showed, you really need a barcode that integrates. So you can simply just do an amplicon PCR of that barcode and look at guide representation. So lentivirus actually has been very difficult to use to make small cell gems. We've always used adenovirus, and the field historically has, because there's just very low penetrance when and a very long latency when lentivirus has been used. So let's just keep that in mind. And so Monty Winslow's lab really pioneered this with the Petrov lab in adenocarcinoma models, looking at KRAS and then looking at cooperative tumor suppressors in a pool that may increase tumor growth. So this is really looking at tumor suppressor genes. So I mentioned the limitations of lentivirus. There are a lot of advantages of lentivirus, but the limitation is that tumors, at least small cell tumors, just don't form that well in lentiviral delivery. But the Sage lab found that pre-treating these mice with naphthalene, which is really the ingredient in mothballs, also present in tobacco smoke, creates this injury and really allows for tumors to form with lentivirus. And in this model, tumors were forming after about 5 months. And so what the Sage lab did is they introduced a whole small guide RNA library. So this is really mostly focused on candidate tumor suppressor genes in small cell lung cancer. But the nice thing is this could be delivered in a pool, and then mice are intratracheally injected. This is in RPR2 mice with Cas9 or without Cas9 as a control, with this virus, treated with naproxen a couple days prior, and then basically looking at the tumors at the end time point. And what they did is and with any screen approach, you want to take the whole lungs here and do sequencing for guide representation. And that's what they did. This is at 14 weeks after intratracheal injection. And what they found really that, consistent with an on-target effect, there was really no effect of any of these guides in the mice that did not have Cas9. So I think that's reassuring. And the mice that did have Cas9, most of the guides were pretty neutral, but there were a couple strong hits, one being a positive control, P10, which we know from work from Tyler Jacks's lab, accelerates small cell tumor formation. But then other candidate tumor suppressor genes, potentially new, TSC1 and Cadmium-6A is a little bit enriched. But we had found actually Cadmium-6A doesn't really have consistent with our findings, it doesn't really have a big effect on accelerating tumorigenesis, more of a plasticity phenotype. So Julian had validated TSC1 as a new tumor suppressor gene. So I think this is a proof of concept that pooled screens can be done using these types of approaches. And we're able to overcome the low efficiency of lentiviral transduction. But I think there's a lot more to do here with thinking about pooled approaches in vivo of how we can more quickly screen genes for growth, or you can imagine other phenotypes. So now I want to switch gears a little bit and talk about allograft approaches. And as I mentioned, everything we talked about to date is either classical genetic models or CRISPR-based somatic editing, but these are all autologous models where tumors form from native cells in the mouse lung. But these approaches, although CRISPR does accelerate the ability to study genes and higher throughput, still have very long latencies and are very burdensome approaches. So there's some questions we can ask that we may be able to answer with some of these allografts. And so this part really has two different parts, kind of standard allografts that we'd use in Black 6 mice, and then these pre-small cell lung cancers or pre-SCs that Quan Park's lab developed as a way to look at what might induce small cell transformation. So first, to talk about allografts. So one of the big advantages is the throughput, right? If you make an allograft, you can inject that same allograft into many, many mice all at once. And then the latency. So tumor formation, as I mentioned, the CRISPR model, 8 or 9 months. In these models, we have models that form tumors as early as really 10 days to 2 weeks. And so several labs have adapted this over the years. I'm just listing some of them here. We first started working on this really in collaboration with Hua Zhang and Kwok Wong when we were first developing this model. And so I think the idea from most of these labs is really the same, is that you isolate tumor cells from a pure background, most often Black 6 mice, but it has to be a pure background mouse, and make either a gem-derived cell line or an organoid. And then you can transplant it back into that immunocompetent mouse of the same background and grow a syngeneic tumor. So these can be, the throughput is much higher, as I mentioned, the latency shorter, and really good, if you want to ask questions about, therapeutic approaches, particularly with immunotherapy or impacts of a treatment on the microenvironment. But as you can imagine, in comparison to an autologous lung cancer, we're injecting these cells at different locations within the mice. And, the easiest thing to do is to do subcutaneous, but there is, caveats of that. And so, the microenvironment is probably quite different than the lung. So different investigators, I'll just mention a couple of the studies here. Hua Zhang and Kwok Wong had developed an intrathoracic injection approach where they could grow these in the lungs to really recapitulate the lung microenvironment of tumors. And then Julian Saja's lab had done intracranial injections to study, how the brain tumor microenvironment may modulate small cell tumor formation. And so another question we can ask with these genetic models is really what drives tumor formation. So I mentioned there's a long latency, no matter what the model, I guess, except for the MYC model, but there's still a couple month latency there. But most of these latencies with RB and P53 are 6 to 9 months. And so Kwan's lab had isolated cells early on from one of these pre-malignant lesions that was null for RB, P53, and heterozygous for RBL2. And the idea there is then you can use genetic manipulations to either knock out one by one different candidate drivers of interest, or higher throughput CRISPR screening, which a nice example of this was in Arnaud Augert's paper in 2020, where they looked at really what would drive transformation at a genome-wide scale. And so the idea is really you can test your hypothesis with these engineered pre-SCs, and you can put these back into immunocompromised mice or immunocompetent mice. And so I think there's a lot of questions. There's much higher throughput, obviously, than making a autologous model, and there's specific questions you can answer here. And so everything I mentioned, I think all of the labs, very collaborative if people are interested, definitely for the pre-SC or TubaSeq or the CRISPR model myself. I think definitely reach out to the investigators that are primarily doing that. And so just to wrap up of the pre-SC models, very tractable for engineering candidate drivers. These can go into immunocompetent mice. These are more like an ASCL1 subtype. It's not really to study tumor prevention though. So just a couple ideas to wrap up. Just some limitations with some of the approaches we're using. Clearly, I think CRISPR mouse modeling with somatic editing has decreased breeding time, but the latency is also still quite long for these models. So I think, the ways that we can study this in higher throughput, such as with TubaSeq, or if there's other approaches, I think will allow us to understand more information or a specific question surrounding a phenotype in higher throughput. I think one thing to point out, just comparing small cell in the mouse to small cell in the brain, small cell in human, is in humans, small cell has one of the highest predilections for the brain. We actually, in the limited stage disease, often give prophylactic cranial irradiation to the brain just because the chance is so high that that patient will develop brain mets. And if we want to cure the disease, often we need to take care of the brain, even if there's nothing that we can see. So I think one limitation of the small cell models is that they don't actually spontaneously metastasize to the brain. I think this is actually quite an interesting question. I don't think the Saj lab has done some work on it, but really no one's really been able to identify models that spontaneously would metastasize to the brain. And then, as I mentioned, we know a lot about small cell transcriptional diversity now, but there's really no good PAL2F3 or inflammatory genetically engineered mouse models that we can use at this point. So some future opportunities. I think, the CRISPR technologies, what I mentioned as far as CRISPR goes are pretty old technologies at this point. Those are kind of standard CRISPR knockout at tumor initiation, but we know that now you can manipulate Cas9 to deactivate its enzymatic activity and direct it toward transcription start sites where you can activate genes. You can manipulate Cas9 to base edit. This is basically to make nucleotide mutations, prime edit, and then Cas12 has other advantages where Cas12 compared to Cas9 is very good for multiplexing guides. And so in the last year or so, actually, most of these mice have become available. And so I think there's, real opportunities to think about if the specific question does involve a amino acid deletion or mutation or substitution, or if we want to model gene activation. I think there's huge opportunities here, and no one's really done that. And I think it will give insights that our models currently cannot give. And then, as I mentioned, most of the models that we use, genetically engineered models, inactivate genes at tumor initiation. But in reality, there's truncal mutations in small cell, and then some genes are probably inactivated later on, and that may have a different phenotype compared to knocking out all genes at once. So thinking about how we can induce or activate target genes at different stages of tumorigenesis, I think will be important. And then, as I mentioned, I think the tuba-seq approach is very useful, but can we think about sort of higher throughput in vivo CRISPR screen? So with that, well, I'd just like to mention, if you want to, a lot of these concepts are in this review that I wrote with other leaders in the field. So I encourage you to go check it out. And definitely don't hesitate to email me if you have questions. So really this talk, I just want to thank Julian Kwan, David Prudy, who we really wrote this whole review together. And this is my lab and some of the collaborators on that KIM6A project that we talked about. So thank you so much for listening. I'll take any questions.
Thank you very much, Matt. It was a really fascinating presentation, and especially for those that are not